So we are happy to introduce the keynote speaker, Prof. Shay. If I will introduce him, I may spend half a day, if not the full day. So I would like to make a shortcut. Please, Dr. Prof. Shay, welcome. The stage is yours.
Since we're running behind, I encouraged him to make the introduction as short as possible so that we could try to get back on schedule. Well, first of all, I want to thank all the organizers for hosting us and for inviting us back. It's always a pleasure to see friends. What I'd like to do today is, we've heard a little bit about personalized medicine, precision medicine. I'm going to go from a very general talk to a very specific topic. Many of you know that I have some other interests, and I've been interacting with many of you here at the Center for Genomics Medical Research. But today I'm going to talk about a project that I've been working on for a long time, which is connecting aging and cancer. And the question is, are the ends of our chromosomes, called telomeres, and the enzyme that maintains the ends of our chromosomes, telomerase, the connection? So I'm going to have a second little subpart, which is a new concept, which is how you can regulate genes by the length of telomeres. And so for you to appreciate why that's an important question, I first have to give you some introductory information. So by way of introduction, I think most people probably know that human cells have 46 chromosomes, 92 ends, and the ends of all of our chromosomes are capped with these elements called telomeres. Telomeres are these T2AG3 repeats at the ends, and they're there to sort of cap the ends of each of our chromosomes. It's actually been known for now over 30 years that, in fact, telomeres progressively shorten every time a cell divides. And it's been believed and has been discussed many decades almost now, over 20 years, that when telomeres get short, that induces some sort of DNA damage signal that causes the cells to undergo a growth arrest, or what they call replicative senescence. This doesn't just happen in cells in culture, it happens in real human beings. So if you look at tissues, reproductive tissues, etc., you can see when we're born or we're young, we have very long telomeres, somewhere on the order of 15 kilobase pairs of telomeres. But as we age, all our telomeres in all of our cells that divide, like the colonic mucosa, blood cells, etc., all get progressively shortened. This is a correlation. It certainly doesn't prove telomeres cause aging, but it's actually a very good biomarker of your, what we would call, instead of your chronological age, your biological age. So what's the problem with having short telomeres? The problem with having short telomeres is that if they get too short, you lose tissue renewal capacity. This is a failure of stem cells to divide in sufficient numbers. You don't run out of stem cells. There are humans that have lived well over 110 years of age, and so we know that those people may be frail, but they still have stem cells. They can still heal a wound on their skin. They can still make mononuclear cells in their blood, etc. There's a more recent consequence of short telomeres, which has been called SASP, or senescence-associated secretory phenotype. And that concept is really very simple. It says that when cells get short, that these senescent-like cells can actually secrete a series of chemokines, cytokines, that can lead to increased inflammation and decreased immune responses. And there's been many correlations over the last decade. There's been literally thousands of papers showing that if you can measure your telomeres in your peripheral blood leukocytes, that short lymphocyte or leukocyte telomeres strongly correlate with the onset of a variety of diseases such as cardiovascular disease, increased risk for liver disease, including aplastic anemia, and of course cancer. So this has sort of led to the idea that what we call the telomere hypothesis of aging in cancer. We now know from literally hundreds, if not thousands of publications, that most pre-cancerous lesions or pre-malignant lesions have very short telomeres. And so we think that, in fact, short telomeres, in fact, may be, at least initially, a cancer preventive mechanisms for organisms that are large, like we are, and that live a long time. And that this may be a sort of a blockade against the initial development of cancer. But obviously, in combination with other genetic alterations, this could drive genomic instability and leading to an increased risk of developing cancer. Now, one of the hallmarks of cancer, most of you have seen and heard about the various hallmarks of cancer, is that cells can divide unlimitless. They can divide forever, basically, or they're immortal. So how do cells, if they have short telomeres, stabilize their telomeres for the long-term growth of the advanced cancer? And the way they do that is by activating this enzyme called telomerase. Now, telomerase is the only cellular reverse transcriptase in our body. It is one of the key hallmarks of cancer, as I've already mentioned. It is actually present in over 90% of all cancers. And in fact, today, there are mutations in the telomerase, the TERT promoter, the telomerase reverse transcriptase. Mutations are the most frequent non-coding mutation in cancer. So we now know that in malignant melanoma, 70% of patients with skin cancer, especially melanomas, have mutations in the promoter of the telomerase gene that leads to a new transcription factor being able to activate this important enzyme. The important thing is that telomerase is actually not present in the vast majority of normal tissues, and that almost all cancer cells do, in fact, have very short telomeres. So when you put all this together, you're sort of asking your question, so would telomerase actually be a good target for cancer therapy? So the first thing that I ask myself is, why is it only in 90%? Why isn't it in 100%? 90% is pretty good. Well, it turns out there are certain cancers, in fact, that don't have telomerase. And the example here is one from a pediatric cancer. This is called neuroblastoma. It's an adrenal tumor, medullary region of the adrenal gland, that occurs in small children. And like all cancers, they grade them by stage 1, 2, 3, 4. And what you're seeing on the left side here is what we call the TRAP assay, the telomerase or the telomeric repeat amplification protocol. And as you can see, with increasing stage, you get more of this sort of banding pattern that you see, which is actually just how the assay works, showing that at higher stages you wind up with more and more of these hexameric repeats that you see as a ladder. You don't see any of this enzyme activity in normal tissue, and of course if you inhibit the enzyme by treating with RNases, it goes away. The thing that was surprising in this study of ours was a category of this pediatric cancer called 4S neuroblastoma. And when you look at that particular cancer, these children are born without any telomerase activation, yet they have metastatic disease. And when you look at their telomeres on the right side of the slide, what you can see here is what we call a TRF gel. This is a terminal restriction fragment. This is one of many ways to measure telomere length. And what you see is that the children with 4S neuroblastoma have very short telomeres. The N stands for normal, T stands for the tumor. So their normal tissues are up there above 10 kilobase pairs, whereas their tumors are very short. So we asked a simple question: what is the prognosis of patients with telomerase negative tumors? And in fact, this is one of the more interesting results, and that is these children that don't have telomerase activity, this is a Kaplan-Meier survival curve, actually have outstanding outcomes. That in fact, if you don't have telomerase, that almost always correlates with long-term survival, whereas those small percent of children that do have telomerase activity, this is a very bad outcome. So again, what this particular example showed is two things. One is you don't need to have telomerase to have an advanced cancer. Clearly, these children without telomerase didn't have cancer, but it's what's perhaps more important. It said without a mechanism to maintain the telomeres, your cells cannot continue to grow. Or what we believe happens in this particular type of cancer is eventually those metastatic cancer cells can't continue to divide. They stop dividing. They're then recognized by macrophages and phagocytic cells in the body, and they're cleared. So we think that this really is a proof of concept that telomerase inhibitors may be a really potent anti-cancer protection mechanism. So what I'd like to do now is to tell you about a clinical trial we've just completed, and then I'm going to tell you about a new approach for targeting this enzyme, telomerase. So the first direct telomerase enzyme inhibitor that went through clinical trials was called imetelstat, or GRN163L. It's really not a small molecule. It's actually an oligonucleotide, and what it does is it recognizes and binds to the telomerase RNA. So to make the enzyme telomerase, you need a functional RNA and you need the reverse transcriptase. And those two have to come together. They have to go to the telomere. So every time the telomere shortens a little bit in cancer cells, this enzyme complex re-elongates the telomeres just a little bit. And so what you wind up seeing in cancer cells is that the telomeres stay short, but they never get shorter. They just stable. So this particular imetelstat is a competitive telomerase template antagonist. So it's not antisense. It doesn't target messenger RNA, but the functional RNA that is required for recognizing the telomeric repeats. It inhibits telomerase and telomere shorten in vitro and in xenograft models. Lots of publications were presented on this work, and therefore it moved forward to a phase 1 clinical trial. It was reasonably well tolerated in patients, and they finally, just this last year, completed a phase 2 trial for stage 4 non-small cell lung cancer, which is now published. This is the concept. The concept on the left is standard of care chemotherapy or radiation therapy. What you see on the left side of the slide is that one is presented with a patient with a reasonably large tumor mass. One then treats that patient with chemotherapy or radiation therapy. You get a partial response, especially with solid tumors, and almost universally you get a recurrence of disease. And this is our number one problem today. That's due to drug resistance, drugs not being effective enough. We could talk about that for the rest of this conference and not come to a satisfactory conclusion. But I think most people agree we need more targeted therapies, more precision therapies, better enrollment biomarkers for patients that are being treated with cancer. The important thing about our slide on the left is we're not doing anything to the telomeres. So we reasoned that if we were to combine chemotherapy with a potent telomerase inhibitor like imetelstat, that not only would we get these partial responses, but during this treatment period we would get shorter and shorter telomeres, and maybe we would have long-term durable responses instead of only a partial response. Well, it turns out in the initial trials there was increased toxicities in a breast cancer trial when you combine standard of care chemotherapy with a telomerase inhibitor. We could discuss why that is. It turned out that patients were getting low platelet counts, thrombocytopenia. And when that happens, you have to go off of drug therapy. And for a thing that is trying to drive the telomere shorter, that was a real problem. So instead, we decided on a different approach. We said, for patients with non-small cell lung cancer, what we usually do in almost all cancers today, we have what we call first-line therapies. These are therapies that have been worked out over the last few decades. And for most patients with non-small cell lung cancer that are not resectable, this is usually stage 3B or 4, that they go through a round of a microtubule inhibitor, paclitaxel, plus cisplatin. This is called doublet chemotherapy. And again, in advanced non-small cell lung cancer patients, 2 out of 3 patients actually do pretty well. Again, this is late-stage metastatic disease. And so what we found was that maybe what we should do is try this imetelstat in the maintenance setting, because all these patients are going to relapse. And so we then designed a phase 2 trial to add imetelstat in a maintenance setting. In other words, in those 2 out of 3 patients that managed to live long enough and have partial responses, they had to be given an additional treatment. And that was the name of the trial, which is listed on here. I won't go through the whole details of this, but in fact, this was basically giving patients frontline or first-line therapy, and sometimes they were on an angiogenesis inhibitor, bevacizumab, and then those patients that did well from their first-line therapy were randomized either to imetelstat alone, imetelstat plus bevacizumab alone, or just observation. Some patients, after going through a first round of therapy, decide that's enough. So that was the trial. There was 114 patients in this trial, and the results are shown on the next slide. So if you just look at the initial results, it looked like at the median survival, there was about a 3-month overall median survival. Now, I'm not an oncologist. I'm a basic scientist, and to me, there are pharmaceutical companies that will go to the bank with a result like that. They say a 3-month median survival, even a slight increase in overall survival, is worth trying to go and to do a phase 3 trial and get FDA approval. The company that was helping to sponsor this trial decided not to pursue this, and I think they made a smart decision for the following reason. If you look at how this drug works, a major problem with direct telomerase inhibitors is really the long lag period. When you add this telomerase inhibitor into a patient, it is starting to shorten those already short telomeres, but the cells don't immediately die. In fact, those cells have to grow for a while, as you see in the blue and the pink on this slide. If you have short telomeres, the tumor is actually going to grow for a while, and eventually a subset of your cells in that heterogeneous tumor are going to get shorter and shorter, and eventually you will have the cells to undergo apoptosis. And so that lag period was a real problem. So we sat down and scratched our heads and said, what we really want is a telomerase type of inhibitor that would work just like chemotherapy, where you'd have a very rapid response. And that's what I want to tell you about. This is all new work. Part of it is published. It was just published in Cancer Discovery earlier this year. What we were looking for, as you see on the right side, is a way of causing cells with telomerase to basically uncap their telomeres so they would be recognized as DNA damage, so the cells would immediately undergo apoptosis. So we sat down and said, what would be the best way to approach that? And the way we approached that was to say, let's see what other drugs are out there, and could we design a drug that might be more preferentially recognized by telomerase and incorporated into the telomeres. And so we looked at one of the original drugs that's ever been used as a chemotherapeutic drug, not the first, but one of the early ones, called 6-thioguanine. And what we did is we said, the telomeres are full of these tracks of G, so it's T2A, GGG, and you have thousands of these repeats. And I said, what if we put into a telomere a nucleoside that would now not let the telomeres be capped properly, and they'd be recognized as damaged. So we modified 6-thioguanine into the compound you see at the bottom right there, 6-thio-2'-deoxyguanosine, or 6-thio-DG. And we predicted that when this was given to cells, it would be preferentially recognized by telomerase. This altered nucleoside would be incorporated in the telomeres, and it would be almost immediately recognized as having DNA damage. And so we tested that on a panel of cell lines. So this is a typical experiment. If you look at the top where the control, this is just a panel of 3 non-small cell lung cancer lines. And if you look at 6-thio-deoxyguanosine treated with low micromolar for 1 week, literally 99% of all cells were killed. We had a panel of over 100 cancer cell lines. And as you can see, out of all the cancer cell lines that are in green that are reasonably sensitive to this drug, only 4 of these cell lines were, in fact, resistant. That's actually a pretty good track record when you think about that all these non-small cell lung cancer lines were mostly obtained from patients with metastatic disease, probably after going through a few rounds of chemotherapy, and I'll come back to that. So the reason why we use this panel of cell lines is because we have complete genomics data on this. We have DNA methylation, we've got exome sequencing, we've got DNA microarrays, et cetera. So if you just look at the microarrays of this is a panel of 71 cell lines. What you can see is these 4 on the left, where you see the red and the green, completely separate on cluster analysis as a heat map. Red means there's these 4 lines that you see on the left completely separate from all the other 67 cell lines on this particular analysis. That suggests that we actually have a biomarker enrollment criteria. We can say if your tumor looks like that on the left, you would be a bad candidate for 6-thiodeoxyguanosine. And I think that's what's really becoming important today in this era of precision or more personalized medicine, is to make sure the drug you give a person has a higher probability of working, and we don't give drugs to people that we don't think will receive benefit from it. So the second question that you had asked, or I asked, was what effect does this have on normal cells? And without going through all the details, these are normal cells. They still look normal after treatment, and you can look at the IC50 charts. On the top is a colon cancer cell line with an IC50 of about 1 micromolar. To the right is a lung cancer cell line, A549, with an IC50 of 2.4. And on the bottom left is just normal diploid primary cultures of human colonic epithelial cells. And on the right, some normal human fibroblasts with IC50s of over 100 micromolar. So this suggests that this drug is not affecting normal cells, but it is very potently killing cells that have telomerase activity. Normal cells don't have telomerase, as you might recall. And then the other important thing, the criteria, was that we suggested that this drug would cause DNA damage specifically at the telomeres. And so if you see this little image on the left versus the one on the right, on the left is a cell that was treated with 6-thiodeoxyguanosine, wasn't treated with 6-thioguanine. And what you're seeing in green is one of what we call the shelterin proteins. This is one of the telomere binding proteins. And the green dots reflect the little telomeres on all of our chromosomes. If you look on the right, this was treated with 6-thiodeoxyguanosine. This is our new telomerase-mediated uncapping compound. What you see is when we co-stain with TRF2, which is the shelterin protein, gamma H2AX, which is a DNA double-stranded recognition, and what you see in yellow is in fact co-localization. So what you see on the right is that a number of the telomeres are actually co-localizing with DNA damage. So what does that really mean? Well, the first thing you got to understand, and let me just show it to you in real data. This is similar thing, but with the data at the bottom. The important issue is that the telomeres only represent 1/6000th of the total human genome. So to see a TIF, as we call it, a telomere-induced foci or dysfunctional foci, that would be very unusual by chance alone. And to see that, in fact, on the bottom right, that after 72 hours of treatment, 35% of all cells in a population have 4 or more of these TIFs, suggests that this is working by the mechanism which we thought. Another important proof of concept is shown here. 6-thioguanine, remember, was the parental compound from which we derived this nucleoside. And so if you look at the top, we're looking at a normal diploid fibroblast, and on the bottom 2 panels, we're looking at the same fibroblast that we put telomerase in ectopically, so we immortalize cells with telomerase. And then we treated these either with 6-thioguanine or 6-thiodeoxyguanosine. So on the top you see, again, it's the same thing, TRF2, the shelterin protein, the telomere protein, the gamma H2X, which is a DNA damage foci. You could see in normal BJs without telomerase, doesn't matter what you treat them with, you don't see any TIFs. But in BJ cells that have been immortalized with telomerase, you only see TIFs in those cells that were treated with 6-thiodeoxyguanosine, not 6-thioguanine. So that was another important proof to us that this drug was working by the mechanism that we thought it was working. And finally, we wanted to know, how did this compare against 6-thioguanine? Because it's still used for arthritis, it's used for inflammatory bowel disease, it's used in some certain pediatric leukemias. So we just took wild-type mice and asked, do in fact the mice lose weight, their toxicities and things like that. And as you can see, while 6-thiol-guanine is well recognized to have certain cytotoxic effects, the animals on the top did lose weight at what we would consider therapeutic doses, somewhere between 1.6 and maybe 2.5 milligrams per kg in a mouse is an effective dose for therapeutic indicators. And as you can see on the bottom, when we went up to 5 mg per kg, all the mice actually died that were on 6-thiol-guanine, but only the animals just didn't gain weight with the 6-thiol-deoxyguanosine. So there's likely to be some side effects of this drug, but it's going to be much milder than what we expect to see with 6-thiol-guanine. And finally, we did xenograph experiments, and it also beat 6-thiol-guanine. And xenograph experiments, as seen here on the bottom, is the 6-thiol-deoxyguanosine. The tumors basically did not progress during the 2.5-week treatment period. We did, obviously, toxicity testing, and I'm not going to waste time going through this, but basically there was less toxicity than what we saw with 6-thiol-guanine, and we didn't see any obvious histopathology from 30 days of treatment. Again, this is using 2 mg per kg. And finally, I want to just say one important thing, and the important thing we heard about earlier had to do with overcoming drug resistance. Without going through the information on the left, a student in John Minna's lab took a lung cancer cell line and subjected it over 18 cycles to exactly the kinds of treatment that you would do for a patient, giving them cycles of paclitaxel and cisplatin. And what she derived, as you see the red lines over there, she could make these cells much, much more resistant to paclitaxel carboplatin. And that's classically what happens. And what we did is say, are those drug-resistant cells sensitive or resistant to 6-thiol-deoxyguanosine? And as you can see on the bottom right, they're still sensitive to the drug. So it looks like this could be a really good drug going forward to overcome multi-drug resistance. And so to summarize this part, basically what we've shown is that, in fact, telomerase still remains a highly attractive target for cancer therapy. For every other targeted therapy, let's just go back to angiogenesis inhibitor, there may be 20 or 30 different drugs that have been either approved or that are in clinical development. And in telomerase, there's imetelstat and this new drug that we're working on. So this is an under-researched area, and we need to have more scientists working on telomerase as an attractive target for cancer therapy. And we showed that this new nucleoside analog of 6-thiol-guanine, 6-thiol-deoxyguanosine, shows both general DNA damage and telomere-specific damage, but only in cells that express telomerase, which is important. It's a low molecular weight, oral available drug. It's less toxic in vitro compared to 6-thiol-guanine, and it overcomes multi-drug resistance, and it's inexpensive to make, and it should be easy moving forward into clinical trials in the future. So that brings me to the last shorter part of my presentation, and that is, what really is the mechanisms that are causing these short telomeres to induce damage or to make cells stop dividing? And the classical idea is shown on the left, which is that telomeres form these special looping structures. They call it a T-loop. And they think when cells get short enough, the telomeres get short enough, that loop gets unwound, basically, and it now looks like a DNA double-strand break. And if you can't cap the telomeres with your sheltering proteins, this is what everybody in the field believes, and I believe it too. But I think there's a second possibility that people haven't been thinking about very much, which we call, on the right, telomere position effects. This concept has been around in yeast biology for almost 2 decades, but hasn't really been pursued very much in mammalian cell biology. The idea is that when you have long telomeres when you're young, there could be genes next to a telomere that are silenced, and as telomeres gradually shorten with increased age, that you could actually be turning on genes that are close to a telomere. And I will come back to that in a minute. First of all, I just want to show you quickly DNA damage does occur at telomeres. So if you look at this is fibroblast when they're senescent, and you look at all these recruitment factors, these DNA repair factors, MBS1, MDC1, et cetera, 53BP1, they co-localize with gamma H2X. So we know that a lot of the factors that are recognizing DNA damage are there. We don't know that those are necessarily always at telomeres. So let's go to the TPE. So we know there's DNA damage, but what is the evidence for telomere position effects regulating some aspect of senescence or aging? So in the yeast field, it's been shown that, in fact, if you take a gene from an area of the genome that's where it's actively transcribed and move it near a telomere, it becomes positionally silenced. So we know there's something special about the chromatin that are near telomeres. And in yeast, it's been shown the strength of TPE is proportional to the telomere length. So if telomeres are shorter, you can get more genes close to a telomere being turned on. And when we got into this field, we asked a simple question: does TPE exist in mammalian cells? And if so, this would be a really elegant mechanism, if you think about it, to actually turn on a gene before you have a DNA damage signal. Because if you could do that, you could start thinking about things that affect people over decades. You know, what turns on the signal to go through puberty in a young child, you know, in a teenager? You say, well, it's hormones. What turns on the hormones? What's the signal, okay? Is it just growth? Or could it be that as your telomeres shorten as a teenager a little bit, some gene gets turned on somewhere else that was silenced prior to that? So we liked that idea, and we decided to spend a little bit of energy to study that. And we did this first with a graduate student in the lab. And we, without going through the details, we wanted to first of all prove that TPE actually existed in mammalian cells. So we made a luciferase reporter. We either put it internally, as you see the red dots on the left, or we inserted it near a telomere, and we could show by looking at the luciferase expression that the internal luciferase reporter was 10-fold higher expression than the one that was by a telomere. So it looks like the same phenomena that was happening in yeast was happening in mammalian cells. But nobody had ever shown an endogenous gene in humans was regulated by TPE. The idea, again, in cartoon form, just so you can appreciate it, is that when telomeres are long, you would have some sort of silencing of genes next to the telomere. As telomeres shorten, gene 1 would turn on, then possibly gene 2. So the way we went after this originally was sort of old-fashioned today, is we actually made what we called a telomere microarray chip. So we made a spotted array containing over 1,300 genes from single copy genes that were subtelomeric within about a megabase or a little bit further. As that turned out to be quite a few genes on the spotted array. We did it in triplicate. And then what we did is we added some other control genes and all that to this array. And then we asked, was there any differential expression if you looked at RNA or DNA from, or RNA from a young cell versus an old cell, okay? An old cell would have short telomeres. Did we see any genes going up in the old cells that was silenced in young cells? And it turns out this is what you would expect, okay? And we had good controls for these experiments. You would expect in a young cell with long telomeres for this gene to be low expressed. As the telomeres got shorter and shorter and you're turning on a gene next to a telomere, it would be highly expressed. And if you re-elongated those telomeres by putting telomerase in a cell, you should get it silenced again. We weren't interested in DNA damaging genes, so the way you avoided that was by having taking old cells and re-elongating the telomeres. To make a long story short, we found a gene called interferon stimulating gene 15 that met this criteria. We found others, but this is the one I'll just show you today. It's published. Basically, if you look at it on the top right, which are Western blot, in young BJ cells, there's very, very low expression of ISG15. And in BJ cells, it says 83 population doublings, you can see that the protein levels go way up. And if you look on the far right of that Western blot, you can see when we immortalize cells with telomerase, it goes back down. And this doesn't just happen in cells in culture. It also happens in real people. This is actually skin biopsies from humans, and you can see as you become older and older, more and more of your cells in your skin express more and more of ISG15. So what is ISG15? It's right by a telomere. It's within 1 megabase of a telomere. It's believed to be an innate immunity gene. It can be induced by interferon, especially type 1 interferon. There's a lot of functions about it that we don't know. So we don't know if it's involved in splicing, chromatin remodeling, stress responses. So it's a big unknown, and we're continuing to study interferon-stimulated gene 15 going forward. But we ran into a problem. The trouble is we did this whole experiment, and now we turned around and we asked a question. What about the genes that were between ISG15 and the telomere? Were they also being turned on with shortening of telomeres? And it turned out that wasn't the case. So all of a sudden, classic telomere position effects wasn't holding up. That there were genes, we just call them X and Y here, that were not being turned on with short telomeres, but ISG15 was. So we had to put on our thinking caps. We had to go back and figure out, well, how could telomere length changes be regulating genes over long distances? And that's what we called it. And basically, we started looking at all the really exciting work that people are doing now, looking at chromosome, what they call chromosome looping, which is that things such as how the DNA is folded up, there are enhancers, insulators, various heterochromatin marks that can change how genes are actually expressed in three-dimensional space. So we went through and we published a few papers on this already using a modified circularized chromosome conformation capture method and 3D co-fish to determine if genes near telomeres are really being regulated by chromosome looping. So this was the proof of principle. Using a completely different cell type, this time we used human muscle cell types, we continued to study ISG15. And as you see in the top, with long telomeres, there's no ISG15 expressed in muscle cells, but with short telomeres, just like in fibroblasts, ISG15 comes up. So we decided we needed a second mechanism to really prove that there was some sort of chromosome-telomere interaction. So we actually developed a subtelomeric probe, which you can see here in red, and the ISG15 probe, which is in green, and did 3D fish right on the cells. And as you can see on the left side, in these cells with long telomeres, you could see the green and the red probes were basically adjacent to each other. 92% of the time, they were either touching or there was no space between them. Whereas when you looked at cells with short telomeres, you saw just the opposite. It was as if as the telomere became unlooped, it was actually moving back into its original position and potentially affecting how that gene was regulated. So in a nutshell, what we found is that classic TPE, while it certainly exists, there's a new modified form, which we call telomere looping over long distances, or TPE-old. And as you can see, that would have a lot of explanatory value going forward. So I'm going to end on one last news story, which involves the telomerase gene itself, coming back full circle from the telomerase, the TERT gene, as I was talking to you about. A very interesting thing occurred to us as we were starting these experiments, and that was the TERT gene itself, the telomerase gene, is right by a telomere. And that was really surprising to us. Why would you put such an important gene next to a telomere? And, you know, people have actually known about this gene for 15 years, and we're still not 100% sure how it's regulated. It can be regulated by transcription, as already said. Promoter mutations are very common. There's good evidence it's regulated by splicing, alternative splicing choices. And there's also evidence that maybe there's epigenetic regulation of it. And so we asked a simple question again: does telomere looping of TERT limit the maximum size or length of human telomeres? Nobody's ever really addressed that question. It's more of an evolutionary biology question. And so the first thing you do today, you go out and look at the databases. Everything's been sequenced, so it's easy to go out there and do that. And when you look at all the higher primates, and you look in the green there, that's where the TERT gene is localized. In humans, chimps, gorillas, orangutans, baboons, rhesus monkey, the TERT gene is right by a telomere, whereas in mice and rats, it's really more centromerically located. So this suggested to us perhaps one mechanism that large, long-lived organisms used to regulate how long their telomeres were was to, once they reached a certain length during embryonic development, because telomerase is on during human development, and in a tissue-by-tissue specific manner to get silence, what silences telomerase during development? And it could be when telomeres got to a certain length, the telomere looping could come over and silence its own gene. Was there any evidence for that? And it turns out it wasn't as dramatic as ISG15, but still very dramatic. That is to say, in young cells, that only 22% of the probes were separated. So again, we made a probe to the TERT gene and a probe that was right below the telomere, and the TERT gene's about 1.2 megabases from the telomere. And what we could see is when the cells got old, more and more of these separated. So that was interesting. It was consistent with the idea, and nobody says you have to have both alleles moving apart. It could just be one. If you have to turn on telomerase as part of cancer, you don't have to necessarily turn on both alleles. So that could be part of the reason why a lot of them are still adjacent in this study. And so we looked further. What else changes with increased telomere shortening in normal cells? And it turns out we looked very carefully at promoter region for DNA methylation changes. And while this is sort of tricky to read, I think you can see that the higher the bars go, the more methylation there is. And if you look at the top graph, that's really a cell with short telomeres, and you can see the entire telomerase promoter is undermethylated with increased shortening of telomeres. We also did one additional experiment. We wanted to bypass senescence, so we knocked down an important tumor suppressor gene. In this case, we knocked down P21. And now we are going to be looking at telomerase transcription. And what we observed is in young cells, if we knock down P21, we do not actually see any increase in transcription. We had very good knockdowns. The first lane is a control. Then we had 2 different shRNAs against P21. But you could see in older BJ cells, when you knock down P21, TERT transcription went up. So what I've told you so far is illustrated on this slide. When cells are young and telomeres are long, the telomere folds over and silences the H-TERT gene. And it turns out that as you get older and your telomeres get shorter, you now enter what we have called replicate senescence, or the M1 stage. And under those cases, the telomere is short, and now TERT is becoming permissive by changing its methylation pattern. And then if you bypass that first stage of senescence, which would be the equivalent of a pre-malignant or a benign tumor, then you start getting increased TERT transcription. So this gene is being so carefully regulated because it's an important, at least initially, tumor suppressor mechanism. And, we have to really think about how this is actually happening. And I just want to end by telling you a little bit about what the mechanism for telomere looping is, okay? We don't know this at all, but an interesting, again, genomics approach has just taught us that if you sequence the entire human genome, what one sees is, in fact, that there are interstitial telomeric repeats throughout our whole genome. So this is a paper that just came out last year by Steve Kozak's group, and he showed there's almost 3,000 TT-AGG repeats, large repeats, short repeats throughout the human genome. Every chromosome has these interstitial repeats. And we asked the question, is it possible that the way that telomere looping is working on the TERT locus, at least, is by the telomeres bending over, finding another track of telomeric repeats, and through some of these sheltering proteins like TRF2, that's making the initial bridge to silence these. And just again, a bioinformatics approach is right close to the TERT gene, within 100 kb, one actually does see large tracts of telomeric repeats. To add value to this, we knock down TRF2, which is one of the sheltering proteins, and you immediately lose the entire capping process. Don't have time to talk about our 3C concept, but again, when you knock down TRF2 on the far right, you see the number of contacts that you have between the telomere and the TERT locus goes down dramatically. Also, as part of our chromosome conformation capture, we showed that there were specific interactions, mostly between the TERT gene and another gene right next to it called the CLPM1L gene. And that's actually quite interesting because when you look at this, you see much more interactions with young cells, the blue on the right, versus the PD70 on the right as well, suggesting that, again, there is a change in the number of contacts. This is a cross-linking, a UV cross-linking approach where you can actually see if there's really interactions between the telomeric region and the TERT locus. And all the other genes that are closer to the telomere, as you can see, are not being affected. And what's interesting is this locus, where these telomeric repeats are, this other gene is right there next to it. And we've just discovered that this gene, this cleft lip and palate transmembrane-like protein, is another TPE old regulated gene. And again, we know almost nothing about it, but it meets all the criteria of that. So I just want to end here by saying that telomere looping occurs in a number of human genes. It provides a mechanism to regulate aging by progressive telomere shortening without the induction of a DNA damage signal. Telomere looping occurs between the human TERT locus and the chromosome 5p telomere. The looping with the TERT locus is disengaged in normal cells with short telomeres. Loss of telomere looping correlates with undermethylation of the TERT locus, and that TRF2 may be mediating the mechanism of telomere looping. And we're trying to finish this all up and hopefully be submitting this off for publication shortly. And I'll just end by thanking Wan-Il Kim, who's the champion of the telomere looping project, and Elgin Member, who is the champion of the 6-thiodeoxyguanosine project. And some of our newest work is now being done with collaborators here and in Tunisia. And it's been a pleasure to present this to you. And if there's time, I'll be glad to entertain questions. Thank you very much.
Thank you so much for Prof. Jerry for his outstanding presentation. We learned a lot indeed. And because we are far behind the time schedule, and the next speaker, Prof. Ali Mubashiri, because he would like to catch his flight back to home, we unfortunately canceled this speech. I will allow not more than 2 questions.
Three.
No. 2.
How about 4? I'll get 4.
Okay, 2 questions for Prof. Shay, and we'll go to Prof. Fornas after that. Sorry for this inconvenience. Please, have a seat.
Have a seat.
Yeah. No question? How come? Outstanding talk.
Somebody better ask a question. I can't So the question is, how many of these sequences are like it? We think this telomere position effect over long distance is going to be a major regulatory mechanism. It may be important in a whole variety of age-related diseases. And as part of our collaborations with our colleagues here in the Kingdom of Saudi Arabia, we are going to try to get the most important ones as quickly as possible. We basically have just been using, basically our second good instincts to pick genes that we think might be important. But in fact, we are now taking a list and going through the literature and asking everything that we've got a list of like close to 5,000 genes. We're in the first 5 megabases of every telomere from the genome to the sequence. And we're asking if any of those genes have anything to do with age-related diseases, whether it's neurodegenerative disease, it could be tissue-specific diseases. So there's a whole variety of possibilities out there. And we're going to be what I would be calling cherry-picking the most important diseases where this could be actually important. I think that right now it's still in the discovery phase. And then the question is, of course, what can you do about it? So if you can turn on a gene with just
shortening of telomeres, what effect does that have on any process? And what we need to do is figure out, can we reverse that? Because it's easy today. One can go in and actually turn on telomerase. There are telomerase activators and things like that. So one can ask the question, does that reverse these types of diseases? It turns out there's a variety of diseases which we call telomeropathies. Telomeropathies are really genetic diseases. These are people that in fact have mutations in some component of whatever maintains telomeres. And a paper we've got under review right now with a group from Riyadh, we've actually discovered a family here in Saudi Arabia that have a specific genetic mutation that is in fact a telomeropathy. It involves it's a law-associated RNA binding protein. The details aren't important. What's important is there's probably a lot of human diseases that may be directly associated with the length of our telomeres. We used to think about telomeres as these little ends on our chromosomes. They're there, they don't do anything, they're repetitive DNA. We shouldn't be thinking about these things. They're not genes. But in fact, these could be important regulatory mechanisms, especially in organisms like humans that live a long time. Recently, people have looked at in larger organisms like the elephant, and they've shown that the reason why elephants is called Peto's paradox. The reason why elephants don't get cancer as much as they should, they're 100 times bigger than a human, yet they don't get cancer any more frequently do. And what about the large giant whales, the bowhead whale? And what they found in the elephant is that elephants don't get cancers because they have extra copies of p53 genes. And in the bowhead whale, they found they had increased copies of DNA repair genes. So what is our mechanism? Why don't we get cancer more frequently than we really do? So we think this could really lead us to some new biology about mechanisms underlying regulation of cancer. Well,
Thanks. Please.
So, most of your experiments were done fibroblasts or
muscle cells, fibroblasts.
What's the information in different tissues? I'm wondering in the brain, do you see telomere shortening through
Yeah, so there are tissues where there's less shortening. So neurons, of course, as you know, are mostly post-mitotic. The glial cells, the vascular endothelial cells turn over in the brain. And so you can't apply. This is the danger we get into in science. We want one universal answer to cancer, one universal answer to aging. I think aging is complex, and if telomere biology accounts for 10% of what normal human aging is about, I think that's just fine. And I think if we, so I think there's a danger in saying, does this apply to all tissues? It certainly doesn't.
What is the pediatric ventricular blastoma? Do you see the same phenomena as neuroblastoma, for example?
So there are pediatric benign diseases we own alike, but they're not as aggressive like you said. And a lot of those also spontaneously regress, just like the neuroblastoma. So there is about 5 to 10% of cancer types we don't understand why people get these remissions. And it's possible at least some of those could be explained because they can't engage the telomerase enzyme to maintain their telomeres, and therefore they senesce.
Okay, thanks. One last question.
Very quick question.
Last.
Is there any link between the telomeres and the current abortion, for example, is there any sort of side effect that was recurrent abortion?
Recurrent abortions? Yeah, we don't know. I think it's close to 50% women that get pregnant have spontaneous abortions even before they know they're pregnant. And we don't understand why there's such a high failure rate of fertilized eggs. So I think it's actually something that might be worth looking at. It could easily be involved in that sort of situation. Okay.
Okay, thank you. Thank you very much, Prof. Jerry, for your outstanding presentation. Now,